Good morning, everyone. Today, our group will be presenting about the ethical concerns of creating transgenic cattle for higher milk quality and pharmaceutical production. First, we're going to take a look into the history of transgenesis, which is the transfer of foreign DNA into the DNA of an animal species. The genetic modification of cattle began a few decades ago, and it was started due to the need to enhance the protein content and nutritional value of the milk that humans consume. Specifically, geneticists wanted to increase the levels of the casein protein in the milk content. Previously, the first attempts at transgenesis occurred through microinjection into mice, which proved to be an inefficient process when it was applied to cows. Recently, nuclear transfer technology was developed that essentially implants new genetic material from a donor into the unfertilized cow egg. However, with the development of these transgenic methods, such as nuclear transfer, comes ethical concerns such as difficulty with parturition or childbirth, fetal loss, and abnormally small or large young, and the health risks that can bring. Here you can see a simplified diagram of how transgenic cattle are produced.
There are three main methods of transgenesis used in cattle. The first method of transgenesis, microinjection, occurs when exogenous DNA is directly inserted into the pronucleus of fertilized zygotes, and then generations would express these genes. This method began to be used in cattle for agricultural and biomedical reasons. However, this method is expensive and has low efficiency. In fact, less than 1% of injected zygotes develop into transgenic offspring. The next method of transgenesis is sperm-mediated. This occurs when exogenous DNA is loaded into the sperm. While this method is easy and doesn't require expensive equipment, it shows varying efficiency. It seems that liposomes have been the most efficient at transferring DNA into bovine sperm. The last and most recent method is cell-mediated nuclear transfer. This occurs when a nucleus from a donor cell is transferred into the cytoplasm of an oocyte with no genetic material. The results show offspring with the same genomic makeup as the nuclear donor cell. This method has many advantages, such as being able to introduce a larger range of gene enhancements and high efficiency. However, the issue lies in the survival of offspring in the womb and newborns.
Proposed modifications of milk constituents that are beneficial to humans. Groundbreaking opportunities for the modification of milk composition have been developed through methods of gene transfer. Targeted mutations of genes may broaden the range of opportunities even more than it already has. Utilization of said opportunities depends upon recognition of the sequences that influence tissue-specific hormonally induced expression within the mammary gland. Having said this, targeted expression may be thus used to modify milk proteins. The following are examples regarding the proposed modification of milk constituents. Eliminating beta-lactoglobulin results in improved digestibility and decreased allergic response, while adding human lactoferrin creates an enhanced iron absorption and protection against gut infections. A decrease in the expression of acetyl-CoA carboxylase had a decreased fat content, improved nutritional quality, and reduced milk production costs. Expression of Ig genes ensures protection against pathogens such as Salmonella and Listeria. And lastly, by replacing bovine milk protein genes with human equivalents, this results in mimicked human breast milk.
Scientists have been able to use transgenesis in order to create animals that produce pharmaceuticals to treat human diseases. The gene for a protein drug of interest can be transferred into another organism that will produce large amounts of the drug, such as cows, due to their large-scale production of milk. Farming is the term used to describe the production of human pharmaceuticals in farm animals. Milk from transgenic cows is a major source of pharmaceutical proteins. Promoters from milk protein genes are bound to DNA fragments containing the coding region of the genes of interest. These desired proteins are then extracted from milk and purified. Transgenic cow milk has been modified to mimic the effects of human breast milk, and some are even able to produce human antibodies in their blood. Transgenic cows can also be used to produce medicines and nutritional supplements. Lysozymes are a specific example of a protein that can be produced in cow milk through transgenesis. Lysozymes are antimicrobial proteins that are naturally found in human breast milk. Lysozymes can help protect infants from bacterial infections. These proteins are typically not found in normal cow milk. However, scientists have been able to use nuclear transfer to increase lysozyme production in transgenic cow milk. This can allow for the production of active human lysozymes on a large scale.
Is it ethical to modify an animal's milk production? A few benefits of modifying cow milk would be improving animal health in calves and allowing them to have a better chance of survival, preventing animal diseases such as mastitis, which is an inflammation of the cow udder, and it has human health benefits such as better milk and helpful pharmaceuticals, as well as encouraging dairy products to process faster. Some worries with modifying cow milk would be having potential health problems with the mother cow producing an unusual amount of protein and pregnancy mortality rates. People and calves would be drinking modified products rather than natural, and there are multiple health risks that could occur with those consuming modified milk, such as allergic reactions.
So how do professionals feel about the welfare of cattle that are created using transgenesis? 466 researchers in New Zealand's AG Research Limited Company were surveyed on their ethical acceptance of a transgenic cattle project. They used 4 moral principles to test their stance. The results showed that their feelings on the moral principles aligned with their acceptance attitudes. Overall, they accepted the project and thought it was permissible to use transgenic cattle to produce medicine for humans.
After having discussed an ethical survey on transgenic cattle, it is time for you to decide how you feel about the topic. I invite you to please consider our discussion questions below. Thank you.